Good day, Junior Tickies. My name is Mr. P. I'm going to be taking you through question 3 of the worksheet when we're looking at DNA and nucleic acids. I've worked through a couple of the videos in this section already, so hopefully you've seen those. If you haven't already, please do get your hands on a copy of the full worksheet of questions, as well as the worksheet memorandum, as well as the theory booklet. It'll definitely help you in your preparations towards your final exams. But let's dive right in here to question 3, where we have a look at question analysis and some exam technique, and hopefully we can get into some good habits when answering questions in our final exams. All right, so let's have a look at question 3. So question 3, pretty straightforward here. Here we're looking at a 6-mark question and a 2-mark question for a total of 8. Here it refers to errors that occur during DNA replication. Okay, so again, we need to understand that the context of this question is DNA replication and not transcription or translation in protein synthesis. I found that this is where most students make the biggest mistakes, is they confuse transcription, translation, and DNA replication with one another. So it's important to understand these processes and then answer the questions in the correct context. So here they talk about errors that occur during DNA replication and how they may lead to different mutations. Okay, so what is a mutation actually? So a mutation can be either point or frame shift. So the department is getting a little bit more specific. Sometimes when it comes to these mutations, we refer to our point mutations. We don't really look at frame shift mutations. We broadly categorize these mutations as chromosomal mutations. And that's typically where we look at things like Down syndrome, okay, with non-disjunction in meiosis, or we look at mutations that are gene mutations, which these are examples, so point or frame shift mutations. But it's typically a base substitution or a base omission that we look at here when it comes to these mutations. So we just need to make sure that we get our context right. So here we're talking about DNA replication and the mutations that result, which could either be the point or the frame shift mutation. So let's not refer to the specifics here of point and frame shift, but rather to gene mutations, okay, which is probably what we're going to have here. So if we look at question 3.1, describe the process of DNA replication, and this is for 6 marks. So we have to be specific, but also we can't be too blasé with our answer. We have to make sure that we start at a point and we detail the process step for step and trying to be as specific as possible. All right, if we look at this question particularly now, so we're describing it and we're going to start at a point. So this is where we have a DNA molecule that will first need to unwind itself in order for DNA replication to take place. After it's unwinding itself, obviously it needs to unzip, but before it can unzip, the weak hydrogen bonds need to break. So the sequence of events are so important here. Okay, so we can't have the unwinding before the unzipping, so it's a process and it has a sequence. DNA molecule unwinds, weak hydrogen bonds break, and we're going to have the molecule unzipping itself. Now, there are certain enzymes that are involved in this process, specific enzymes which we don't really need to name. So what I'm going to do is I'm not going to mention them, because if you, for example, mention the incorrect enzyme with its function, you're going to lose marks. But if we refer to the general term, enzymes controlling these actions, you will still receive your mark for what is actually happening, because here we're describing the process. We're not actually asked for the functions of the enzymes. So why do we put it in there if it might cost us marks, especially if we can't remember the detail, particularly when it comes to those enzymes. So here what we're going to do is we're just going to refer to the process of DNA replication. So the molecule unzips, and this we can say is done under the action of enzymes. We don't need to label or list them. What we do now is we take it a step further and we say that because this is DNA replication, both of my strands, because a DNA is a double helix, there are 2 strands joined together. So both of these strands act as a template And this is for our DNA nucleotides to arrange complementary to the bases that are now exposed to the nucleoplasm. All right, so my DNA nucleotides form the complementary strand, and what I have at the end of the day is A joining with T, complementary, and C with G, also complementary, and this is going to result in my two genetically identical DNA molecules. Okay, all right, because after or during cell division, these molecules are going to be pulled apart to form my genetically identical daughter cells. But here we talk about the resulting in two genetically identical strands, and there we have DNA replication detailed step for step. Now my question to you is, do you think that what we've written here is worth 6 marks while there are 8 points? So somewhere along the line there, I'm very, very sure we will get our 6 marks. Even if we've omitted 1 or 2 details that the memorandum requires, I believe we've written enough there for our full 6 marks. Right, let's have a look at next question, 3.2. Here, describe how an error in DNA replication may lead to a gene mutation. And this is where we have the incorrect base in place in the sequence. So where I should have a C, I have an A, a T, or a G, and this is going to cause a sequence change in the bases. Now, what happens now is when we've got protein synthesis taking place, so now I'm talking a few steps ahead now, my amino acids that are generally going to find themselves in some sort of a sequence to form a protein or a polypeptide, my sequence will change because my base sequence has changed. So this at the end of the day is going to result in a different protein, or a protein that is not going to function quite the way that it should function. All right, and this is where we have our gene mutations affecting the functioning of the body to a certain extent. All right, a little bit forward, this sequence change, as I said before, results in the amino acid and then obviously the incorrect protein being formed there. So the incorrect amino acid in the incorrect place in our protein. All right, Junior Tsaki, so a little bit of, let's say, exam technique here, but also a little bit of information, so a little bit of theory involved here as well. So we've gotten through quite a bit here. So these are the questions you sort of got to ask yourself when answering these questions is, what am I trying to communicate here? What is the question actually asking me for? And I think we've done a bit of question analysis and we've answered exactly what the question is looking for from us here. So Junior Tukkies, again, I'm Mr. P. Thanks for joining me for this video dealing with question 3. I hope it has helped you. Looking at, like I said, exam technique, it's very, very important to do our question analysis. And again, if you haven't received or you haven't downloaded the worksheet with all the questions, the worksheet memo and the theory booklet, do so. It's just going to help you in preparing for your final exams. Okay, this is Mr. P signing off. Hope to see you again soon.